So we know that the plastids, they vary according to the color, right? And then there is the colorless plastid, which we know as the leucoplast, right? Furthermore, the leucoplast has been divided into the types such as the elioplast, eluroplast, and amyloplast, isn't it? Now we'll be talking about the chromoplast. Okay, now see. The chromoplast, just like how the leucoplasts were colorless, right? Now we're going to talk about the chromoplasts. Now these chromoplasts, they are the ones that have almost all of the colors except for the color green. Okay, they don't have the color green. So we now talk about the chromoplast. Now remember that these chromoplasts, they can be yellowish, they can be orangish, and apart from that, they can be reddish in color because of a particular pigment which is in abundance in them, and that is the carotenoid pigment. Okay, now they are either formed from the leucoplast. Now those leucoplasts that get color in them, they are they become the chromoplast, or those chloroplasts that lose the color in them, they become the chromoplast. So they are either formed from the leucoplast or the chloroplast. Now talking about them, just remember that whenever you talk about the chromoplast, so the colors that are dominant in them are yellow color, orange color, and reddish shades. Clear? All right, so in the chromoplast we can write down that these plastids are yellow, orange,
Ma'am,
yellow. Yes.
ma'am, can you please tell me what all are the important diagrams for cell, the functional unit of life?
Yes, yes. I will tell you. See, today, right now it is 9:10 in India. So today for the batch, the class will go on for only 45 minutes. As Jobita has her exam for tomorrow. So I will leave all of you at 9:45, like after 45 minutes or 40 minutes. But for Jobita, I'll carry on afterwards. So in that, I'll tell you all of the tips and tricks. Okay, because there are a lot of topics left. We can't complete all of it in one day, but I can tell you the most important stuff so that you can learn that. Okay. And this chapter is coming only, right?
Yes, ma'am.
So great. Okay. So the chromoplasts, they are yellow, orange, or reddish in color because of the carotenoid. These ones are the most dominant pigments in the case of chromoplast. Okay. Now, have you even seen the tomato that are yellowish in color? Have you seen yellowish kind of tomatoes? Now, you might have seen that after a while, the yellowish tomatoes become pure red in color, right? So the amount of carotenoid pigment starts to increase in them. Or you can say that the orange color, you have seen carrots, right? The carrots are orange in color mostly. So that orangish color in the carrot, it is because of the chromoplast which is present in them. Clear? Any doubts? Yes, Anna?
Ma'am, in tomato, the top part, right, that would be in greenish color. So that's also because of carotenoid.
Hmm. And mostly, okay, apart from that, you might have seen, like, see, as I told you that the chromoplast, they either transform from the leucoplast or from the chloroplast, right? So initially, the tomatoes that come along, they are greenish in color, very starting. Okay. So in that time, they have a lot of chloroplast in them. But slowly, slowly, you'd see that they turn to a little orangish shade, a little orange, yellowish shade, and then finally to the red shade. So what happens in the tomato? The chloroplast, they transform themselves into the chromoplast. Okay. So that transformation happens. So chromoplasts are formed in them. Clear, students? So we'll write that they are formed. They are formed either from leucoplast or chloroplast, meaning either from their transformation these chromoplasts are formed. So they are right in the middle. So leucoplast can also turn into chromoplast, and the chloroplast can also turn into the chromoplast. Now you write down one such example to justify this statement, that change of color from green to reddish in tomato. The most famous case is in tomato during ripening, right? Mention that also, like the tomato does not just change its color. During ripening, the color change happens. So during the process of ripening, and even in the case of chili also, the chili. Okay. So both of these cases, it happens due to transformation. Like, see, the green chilies and the red chilies that are there, it doesn't mean that the red chilies are different. The green chilies only, they turn into the red chilies because of the transformation process. You getting that, students? So this is due to the transformation of chloroplast to chromoplast. Clear? This much is clear to everyone? Also, you can mention one more point, that the orange color of carrot roots is due to chromoplast. How many of you have studied morphology of flowering plants? Like, you've had that done in your school? So you people remember that carrot is a root modification, that it is the storage of food in the root. Hmm? Okay, good. So chromoplast is clear? Any doubts in chromoplast?
No,
All
ma'am.
right. Okay. Now we'll talk about the chloroplast, the one that we are so obsessed with, the one that is the most, you can say, holds the most prime importance in terms of botany, is the chloroplast. Now we know that they are green in color. Why? Because they contain the photosynthetic pigments. So they are greenish. So they are greenish plastids which possess photosynthetic pigments. Photosynthetic pigments, that is chlorophyll majorly, right? This is the pigment, chlorophyll. Because of the presence of chlorophyll, the chloroplasts are green in color. But it doesn't mean that only chlorophylls are there. Apart from chlorophyll, carotenoids are also found in the chloroplast. Have you people done that experiment wherein the separation of color is done, chromatography? Have you done that in school? Okay, maybe you will get to do it. In that, you will see how many things are present in the chlorophyll, or you can say uh, how many pigments or various shades that you can find. Anyway, so talking about the chloroplasts, they are greenish plastids and they have the photosynthetic pigment, that is chlorophyll and carotenoids. Now remember that the chloroplasts are extremely important as far as the photosynthesis is concerned, right? Because these are the only cells in the plant's body that would contain the chlorophyll pigment, and without chlorophyll pigment, photosynthesis will not take place at all. Now, why do you think, students, what is so special about chlorophyll? What does it do? Hmm? What is the function of chlorophyll?
It helps in photosynthesis.
How? You're right. How?
By trapping light energy.
Very good. It traps the light energy. That is the prime importance, because we know that photosynthesis will take place in the presence of sunlight. But what is the use of sunlight if there is nothing to trap it, to catch the sunlight particles? So these chlorophyll molecules, they absorb the light energy and then they turn it into chemical energy so that plants can use it. Okay, now in terms of the size of the chloroplast, so they have a length of about 5 to 10 micrometer. 5 to 10 micrometers. Now talking about the shape, so the shape also is so variable. The shape of chloroplast is so variable that you will find them spherical. There are spherical chloroplast also. Apart from that, there are ribbon-like chloroplast also. Okay, they are lens shaped or discoid chloroplast also. So oval, discoid, discoid means disc-like structure. So all of these are the shapes in which the chloroplast exists in. Now we have talked about the dimension, we've talked about the shape. Now talking about the number of chloroplasts. Now again, the number of chloroplasts depends upon how much is the need of photosynthesis. For example, if we talk about chlamydomonas. Chlamydomonas is an example of which type of organism, students? Chlamydomonas. Is it a bacteria? So there is one per cell in the case of chlamydomona. Is it a bacteria? No, it's not. What is chlamydomonas? Be specific, because we have studied plant kingdom, right? What is chlamydomonas? This is very bad. Tell me what is chlamydomonas. It's a green algae. It's chlorophyce. Okay. So it's a green algae, but it is microscopic. It's very tiny. So there is only one chloroplast per cell in the case of chlamydomonas. And if we talk about the angiosperm, or particularly in the mesophyll cells, so there are 20 to 40 in the mesophyll cells. Clear? So the number varies. Mesophyll cells are present in the mostly in the angiosperms, meaning the higher plants, right? The flowering plants. So in them, the number of chloroplasts will naturally be much more. Any doubts up until now, students?
No, ma'am.
Okay. Now we'll talk about the structure of chloroplast. Now the chloroplast is somewhat similar to mitochondria, and it is a double membranous structure. Okay, double membrane is present in it. Okay, two membranes, one is the outer membrane and one is the inner membrane. Now apart from that, what happens is that this particular chloroplast, it like the inner membrane encloses a space, just like how in mitochondria there was cristae, right? Same way, in the case of chloroplast, the space is enclosed by a structure that is known as stroma. Okay, so it is a double membrane structure, meaning outer and inner membrane is present. Okay, now moving forward. Abir, why are you late?
Sorry, ma'am. I had to pray Maghrib. It's exactly at 6:30.
Okay,
From I'll prepare earlier and.
okay, okay. All right. So, yeah. So the structure of the chloroplast, it is a double membrane structure. One is the outer membrane and then there is the inner membrane which is present. Now, remember that the inner membrane encloses a space and the outer membrane is continuous. and the inner membrane, it encloses a space called as stroma. Stroma is the space which is enclosed by the inner membrane. Now this stroma, it contains multiple coin-like structures that are known as thylakoids. So thylakoids, this is very important. I mean, mostly the chloroplast. Many a times you'll find questions on like write short notes on chloroplast. Okay, so chloroplast is very, very important. Okay. So when we talk about the chloroplast, so inside the stroma, thylakoids are present. Okay, so thylakoid are the membranous sac-like structures. Membranous sac-like structures. Sac-like structures present in the stroma. Okay, and what happens? They are sort of this like these structures. Okay, and then these thylakoids, when they are stacked upon each other, just like how if you put coins over coins over coins, you make a stack of coins. Same way, when you make a stack of thylakoids, this structure is known as grana. Grana is the stack of thylakoids. Okay, students. So the stack of thylakoid is known as grana. Now, what is the function of grana? See, all of these structures that we have discussed up until now, they are very important in terms of photosynthesis. Okay, they're extremely important in terms of photosynthesis. Now, apart from this, remember that each of these thylakoids, or you can say each of the grana, like several grana are present like this. This is one. Okay, then this is the second one. And each of them are actually connected to each other like this. Okay, and they are connected by a structure which is known as the flat membranous stroma lamellae. This structure is known as stroma lamellae. So remember that the structure which connects two grana together or several grana together, they are known as the stroma lamellae. So you write that the thylakoids of different grana, the thylakoids of different grana are connected by flat membranous tubules, and those tubules are called as stroma lamellae. They are called as stroma lamellae. They are responsible for connecting the thylakoids of several different grana together. Any doubts up until now, students?
No, ma'am.
Okay, very good. Now see, the stroma of the particular chloroplast, it contains several enzymes. And apart from that, now comes the most interesting thing. Apart from containing several enzymes, they also contain their own double-stranded DNA. They also contain their own 70S ribosome. So again, just like mitochondria, they are also semi-autonomous, right? And what is the meaning of semi-autonomous, students? Who can tell me the meaning of semi-autonomous? Anyone can tell me.
They have con they have the things which have cells have separately, and they have their own DNA and ribosome.
Yeah, but like, okay, they have their own DNA, they have their own ribosomes, but how does that make the chloroplast and mitochondria semi-autonomous? Like, what is the meaning of semi-autonomous?
They can make their own protein.
They can make their own protein. They can do their own replication part also, right? So they don't need the cell to do all of these things for them. They can do it on their own. That is why autonomous what? It means self-regulatory, right? A thing that can regulate its own self. So chloroplasts, just like mitochondria, are semi-autonomous. Semi-autonomous cell organ. Clear to everyone? Now, apart from that, remember that whenever we talk about anything that is semi-autonomous, it means that double-stranded DNA is present in them and 70S ribosomes. Always remember, it doesn't mean that they are eukaryotic cell, so they'll have 80S. No, 80S ribosomes, they are present in the cytoplasm. We have studied about it, right? One ribosome is made up of how many subunits, students?
Two subunits.
Hmm?
Two subunits.
Two subunits. What are the subunits? Larger and
30S.
smaller. What? What did you say?
30S and 30S.
Yeah. 70S is the prokaryotic ribosome, right? Mostly. And 80S is the eukaryotic ribosome. Now, 70S and 80S, both of them, they are made up of two subunits. One is the larger subunit and one is the smaller subunit. You know, when you said that today, I recall one thing. See, I was teaching a student about motility. Okay, cell motility. So I asked him, what is like what do us humans use for motility? So I wanted to hear legs, right? We use legs for movement and our legs are made up of bones and muscles, right? But he said car. I was like, okay, very practical, but yeah, he's right in one way. For motility we do use that, but as humans we use legs. So when you answered that, it made me recall that. Okay, you people understood everything up until now? Any doubts?
No.
All right. Okay. Now you people do one thing, you note down till here, then we'll move forward. You people had to write down from amyloplast or elioplast. Which one?
Ma'am, we wrote them.
All of them? Alluro till alluro, all of them. Okay. Okay, you people can switch off the videos. Write down comfortably.
Noted, ma'am.
Everyone's done?
Yes, ma'am. Done, ma'am.
Done.
Done, ma'am.
Everyone's done?
Yes. Done, ma'am. Ma'am, can you Noted, ma'am.
All right, everyone's done. All of you are done, students?
Yes, ma'am.
Okay. Okay, students, so we'll keep it till here only. Now, see, all of you, except for Jobita, you people can leave since Jobita has a test tomorrow. So, like, I'll be helping her a little bit in the extra chapters other than this chapter, okay? So, the rest of you can leave the class.
Thank you, ma'am.
Okay, good night, everyone. Okay, Jobita, are you there?
Yes, ma'am.
All right. So in terms of the chapter that is cell, okay, in this chapter, the sectional view of chloroplast is very important. The chloroplast in general is very important because a lot of questions can be asked from that. Okay.
Okay.
And there is like a diagram which differentiates between animal cell and plant cell.
Yes, ma'am.
You can practice that diagram and also the labelings of the diagram.
Okay.
These two are very important, and the other ones you can just give a look at it, but it's not extremely important. Okay, these are the ones that will be like most importantly asked. Now remember, if they talk about nucleus, remember that Robert Brown discovered the nucleus.
Okay.
Robert Brown. Okay, Robert Hooke discovered the cell. Robert Brown discovered the nucleus. Okay. Now the next chapter apart from this, which chapter is coming? Photosynthesis. Biomolecule. Okay, and
Cell division
hmm,
and plant physiology.
the whole entire thing.
No, plant, 3 chapters: photosynthesis, respiration, plant growth, and development.
Okay. Now see, when we talk about biomolecules, see in biomolecules, have they taught you the chapter biomolecules?
Yes, ma'am.
Okay. So in biomolecules, the most important topic, mostly the question can come from the structure of proteins. The one primary, secondary, tertiary structure of protein. You can look at that also. Okay, and apart from that, you can also look at the enzyme part, the naming of enzyme and everything which is there. Okay, and do one thing before tomorrow. Like your exam is at what time tomorrow?
7 a.m.
7 a.m. Okay, before that, you do one thing with your NCRT book. You read all of the chapters. I am sure that you have learned everything, right? Because tomorrow is exam only. Yeah. So you read all the chapters from NCRT because that will help you to, sort of revise every single thing. And apart from that, from cell division and yeah, the difference between mitosis and meiosis. That is important. Okay, the difference between mitosis and meiosis. And in meiosis, the prophase has been divided into further stages, leptotene, zygotene. You can look at that also. Okay, and the functions, like they say that what are the uses or the functions of mitosis and meiosis, that also you can pay attention to, and all the names, like in what stage of cell cycle what happens, that can also be asked. Okay. Okay, now when we talk about the plant physio, so in that particular chapter, if it is about photosynthesis, in photosynthesis, the photosystem, there is a difference between photosystem, right? Photosystem 1, photosystem 2. That is important. And in that also, they have given like where does the dark reaction take place, light reaction take place, that. Okay, and maybe a question like this can come, like why, like what is the meaning of dark reaction? Why is dark reaction called as dark? Does it happen in the dark? So no, it doesn't happen in the dark, but it is dependent on the products of light reaction, isn't it? That is why it is known as a dark reaction. Now in photosynthesis, yeah, factors affecting photosynthesis. that part, the last part, the law of limiting factors, like how water concentration, temperature, pH, all of these things affect the whole entire photosynthesis, that you can pay attention to. Now, when we talk about respiration, in respiration, see, I am just telling you the topics of these chapters that are very famous, right? The topics that are asked a lot, but it doesn't mean that the rest of the topics will not be asked. You never know, all of those can also be asked because it depends on your teacher, but I'm telling you the topics that are very famous. Now, talking about respiration in plants, in that particular chapter, the whole entire glycolysis process, okay, and the fate of pyruvic acid, that is like what happens to pyruvic acid, the aerobic respiration, the anaerobic respiration, and everything. You should know about fermentation also. Okay. And also remember one more thing, that in those alcoholic beverages that are prepared at home, or prepared by humans, their alcoholic concentration will never exceed 13%. It will always stay till 13%. Okay. Why? Because after 13%, the yeast poison themselves. Okay. So for those alcohols that are sold in the market, they are concentrated, or you can say the alcoholic content is increased in them by a process known as distillation. So after fermentation, distillation happens. Okay. So that is for respiration in plants. Apart from that, in respiration, you can also look at that. The respiration, aerobic respiration part is there. You don't have to learn every single name of the cycle, but you should know what aerobic respiration is.
Ma'am,
Okay.
should we study schematic representation of electron transport system, the table, or just the paragraph?
The paragraph is enough. You can study the paragraph. You can like know everything that is there in the whole entire ETS. Okay, now plant growth and development. See, in that particular chapter, all of the hormones that have been mentioned, the functions of the hormone is important. Like which hormone is used to do what. Like some of the hormones are used to increase the stem length, some is used to increase the root length, right? So all of these things you can. And like, do you have past papers also of your like
No,
here?
ma'am, last year they took humans half yearly human portion.
Okay, okay. So
So
this year
they
they're
don't
taking plant.
so they don't have
Oh,
anything about plant physiology.
okay, so they just have about human physio, and they have cell cycle and biomolecules?
No,
No.
only
Okay.
for us this year they took.
Okay, okay, it's okay. And if your preparation is good and if you revise very nicely, you still have the night, right? So if you revise all the chapters, read everything from NCRT. Nothing out of NCRT will ever come, isn't it? So read everything. Now in plant growth and development, pay attention to all the hormones and their functions. What like auxin is used to do, what gibberellin, cyto, and remember that this thing, the abscisic acid.
Okay.
Abscisic acid is known as the stress hormone. Okay, because it does not do any growth promoting activity. It always retards or slows down or takes the plant towards old age. Okay, so these are most of the topics that you should like pay more attention to. But apart from all of these topics, once you're done, then read the whole entire chapter, NCRT. Okay. Hmm. Okay, so is there any topic that you have not understood that maybe I can like teach you here? Any topic that you want
Ma'am,
particular?
biomolecules is a little bit tough for me.
I can understand that. Yeah, it is because it's all about chemistry in biology, isn't it? So which part?
Polysaccharides.
Polysaccharide. See, polysaccharides, they are sugars only. Okay, but they are macromolecules, meaning they are larger biomolecules, and their monomers, they like each of remember that always a polymer is made up of several monomers, isn't it? So when we talk about polysaccharides, so the polysaccharides, they are made up of thousands and thousands of glucose, or you can say monomeric unit. Okay, each of the polysaccharide. Now, apart from that, remember that in our food, the food that we eat, polysaccharide is the most amount in our food. Clear?
Yes.
Now, you only tell me what are some of the examples of polysaccharides? The most common examples of polysaccharides. It's very, you can say, very naturally occurring. Tell me.
Cellulose.
Cellulose, right? Cellulose, which makes up the cell wall, isn't it? So cellulose is made up of what? Like cellulose is a polysaccharide, so it is made up of how many monosaccharides, or what is the monomeric unit of cellulose?
One.
Hmm?
One.
What?
Only one type of monosaccharide.
Correct. One type of monosaccharide. Apart from that, have you heard about starch?
Yes, ma'am.
Yes. What is starch? Starch is also a polysaccharide, and where it is the reserve food material of plants, isn't it? And starch is made up of glucose. And what happens? It is what we eat. When we eat the rice and stuff, this is what is the energy giving food for us, the starches. So starch is one of the most common type of polysaccharide. Now remember that the starch, it consists of two components, amylopectin and amylose. Two components are there in starch, isn't it? Now you see what is the function of amylopectin and amylose. Amylose and amylopectin, these are the components of the starch molecule, and one of them, amylose, is water soluble, whereas amylopectin is insoluble in water. 20%, I think 15 to 20% of starch is made up of amylose, whereas 80% of starch is made up of amylopectin. Okay. Other than that, see, in your book, there will be given the linkage. See, in the case of amylose, C1 to C4 glycosidic linkage, meaning in the starch molecule or in the amylose component, there is glycosidic linkage present. C1 meaning carbon number 1 to carbon number 4. But if we talk about amylopectin, so it is present, like both the bonds are present. C1 to C4 is also present, and C1 to C6 is also present. Okay. So this is about starch. Starch is an example of a polysaccharide. Apart from that, there is cellulose. Again, a very common example of polysaccharide. This is used to make the cell wall in plants, isn't it? So it is very abundant, and remember that it is the most abundant in the plant kingdom, because all the plants have their cell wall made up of cellulose. Then there is glycogen. Just like how starch is the reserve food material for plants, glycogen is the reserve food material for animals. Okay, it is very similar to starch, but remember that glycogen is for animals and starch is for plants. Clear? What else other than this? You like the structures of protein are clear?
Yes, ma'am.
Okay. And proteins, what is the monomer of protein? The proteins are made up of?
I mean,
Amino acids. Yeah, you're right. Amino acids. Okay. Now, about the amino acids, you know that there is NH2 group present in it and COOH group present.
yes.
And remember one more thing, they can ask you about Zwitter ion also. Like, what is Zwitter ion? So, the amino acid, it can exist as a negatively charged and a positively charged molecule together, right? NH2 gives the positive charge, sorry, yeah, and COOH gives the negative charge. So, because of it, it becomes a Zwitter ion, right? Okay, what else about apart from this? Yeah, and see, when you're studying about the structure of proteins, remember the examples. like the linear structure, what examples are present. Okay, just like quaternary structure, the fourth kind. Here, hemoglobin is one such example, isn't it? So that part you can also read. And see this paragraph, that is denaturation of protein. Remember that whenever we take protein and we put it at a high temperature, the structure of protein becomes damaged. Okay, and the activity also loses. So that process is known as denaturation. Clear?
Ma'am, in cell cycle and cell division, mitosis or meiosis is more important.
Both of them are actually very important. Mitosis and meiosis both. But yeah, another thing, in that starting you might have learn about interphase, S phase, G1, G2 phase. So in that you see what happens in G1 phase, then what happens in S phase. All of these are very important, like what all things take place. Okay.
Ma'am, and diagrams, mitosis.